Can somebody please confirm you can hear me?
Can confirm.
Thank you. So Lab 4, is that what I just saw? Yes. All right, before we begin, are there any questions? Let me share my screen. Oh, my program is still running for the lecture, so my computer is going to be slow for the moment. Let me share my screen here. You should be seeing my screen now.
Yes.
So today is April 22nd, and we're supposed to be covering Lab 4 stains. I do have Lab 2 and 3 graded, so they will be, for the first time, so they will be turned in for the second grading this Saturday. And I have not checked to see how the class voted, so I don't know. I imagine the class voted for 2 gradings, but I don't know. I'll have to take a look. I haven't checked yet. Let's see. Let's start with Lab 4. Are there any questions before I begin? There we go. Lab 4, staining techniques. We're going to discuss simple, differential, and special stains. There is some reading in the textbook. About 2 pages on the size, the shape, and the arrangement of bacterial cells. About 2 pages on the bacterial cell wall. A page or less on the simple stain. 2 pages on differential stains. A page on the Gram stain, and a page on the acid-fast stain. And then we're going to talk about special stains on page 67 to 68. And we'll talk about the negative staining for capsules on page 67 and 75 to 77. The endospore stain on page 68 and page 92 to 94. And then the flagella stain on page 68, as well as page 77 and 78. There are some supplemental materials in today's lab. There are some video clips and another virtual lab. Your learning objective for today, prepare bacterial smears for staining. Understand the principles and procedures used when performing a simple stain, a differential stain, and the special staining techniques. And 3, interpret the results for the simple, the differential, and the special stains. And then define the following terms. What's a simple stain, a differential stain? What are special stains? What's a coccus, a bacillus, a spirillum? What's peptidoglycan? What's a gram-positive cell? What's a gram-negative cell? What's an acid-fast stain? What's the primary stain? What's a mordant? A decolorizer? A counter stain? What's a capsule? What's an endospore? And what's a flagella? Any questions about any of that? Okay. Bacteria are essentially transparent. Therefore, it is necessary to stain them before they can be observed under the microscope. Before staining, you have to make a bacterial smear. A bacterial smear is something you make before you begin the staining process. Students often get that wrong. A bacterial smear is something you do before you begin the stain. To make a bacterial smear, you simply add a drop of sterile water placed on a clean slide. You then add a little bit of bacteria to the water. The bacteria mixed in the drop of water, and then the drop is smeared over the surface of the slide. The cells are allowed to air dry. The organisms or the bacteria are then fixed onto the slide by gentle heating. You need to air dry and heat fix the cells on the slide so that the cells will remain on the slide and they will not be washed off during the staining process. And it looks like the lecture file just finished. And so my computer will start working a little faster now. All right, a simple stain is simply a technique using one dye to stain the bacterial cells. You'll note I crossed out the word basic there. Doesn't matter if it's a basic dye or an acidic dye. You use one dye and that will be a simple stain. You can get the shape and the arrangement of the cells, but all the cells will stain the same color with a simple stain. Differential staining technique uses two or more dyes to provide additional information about the cell. And then lastly, special stains are used to dye a particular bacterial structure, such as the capsule, the endospores, and the flagella. So a special stain is a stain to show you one structure of the bacteria. When we're talking about bacteria, it's important to realize that they have a shape and they have a cellular growth pattern. Here we're looking at the shape. These are all cocci, and that's basically spheroid cell. And then these are different growth arrangements or cellular growth arrangements or cellular arrangements where the cocci are in strings called the streptococci. Here the cocci in our cluster, like staphylococci. The cocci here in tetrads, 4 cocci hooked together. And here the cocci are in doublets, and that's called diplococci. Bacilli and cocci are two bacteria shapes. Coccobacilli is also a bacteria shape. Spirilla is also a bacteria shape. And then some bacteria are pleomorphic, where the shape of each individual cell will vary, and we say they're pleomorphic. I'll show you a better picture of a pleomorphic species later in the class. I'm not sure if it's today or later. The pleomorphic bacteria often do not have a cell wall. So it's the cell wall that determines the shape of the bacteria. And they're pleomorphic because they don't have a cell wall, so they take on many different shapes. Any question about any of that? All right. There are two differential stains, the Gram stain and the acid-fast stain. We're going to talk about each of them right now. The Gram stain was invented in 1884 by Hans Christian Gram. You don't need to know the year, but you should know it was invented by Dr. Gram. And so when you spell it, it should be with a capital G because it's named after Dr. Gram. He devised the Gram stain as a differential stain to separate the patient's cells or differentiate between the patient's cells and the bacteria cells. Microbiologists then began using his stain and they realized that it differentiated bacteria into two very large groups, the gram-positive cells, which stain dark purple, and then the gram-negative cells, which stain lighter pink colored. It distinguishes the various bacteria according to their ability to retain the certain dyes. Crystal violet is the dye being retained by the gram-positive cells, and we'll talk about how the cell wall determines whether the cell is gram-positive or gram-negative. If the bacteria can retain the crystal violet, they're gram-positive. If they do not retain the crystal violet, they are gram-negative. And here we're looking at the Gram stain, seeing a gram-positive rod, gram-positive cocci right there, a gram-negative rod or bacillus right there, a gram-negative spirillum there. The cell wall composition of gram-positive bacteria is structurally different from the gram-negative cells. Gram-positive microorganisms contain a thick peptidoglycan layer, while gram-negative bacteria have a thinner peptidoglycan layer. So here we're looking at the gram-positive cells, and there are the gram-negative cells. The gram-positive cell have a lipid bilayer called the plasma membrane, and then the cell wall is essentially one thick layer of peptidoglycan, and it's just layer upon layer, at least 30 different layers up to several hundred layers. And there you can see some of the layers of peptidoglycan in this cell, meaning right there's, it's hard to see it now, but that's one layer of peptidoglycan there. There's another layer there. There's another layer there. And that's all there is, essentially, in the cells. Peptidoglycan in the cell wall, essentially peptidoglycan. There are other molecules, but peptidoglycan is by far the most numerous. Excuse me. The most numerous molecule in the cell wall of the gram-positive cell. The gram-negative cell wall, on the other hand, is fundamentally different. It does have the cell membrane being a lipid bilayer, phospholipid bilayer. It does have peptidoglycan, so there is a layer of peptidoglycan. And then outside that peptidoglycan, we have more lipids, as well as some carbohydrates. This is a thinner layer of peptidoglycan. I should warn you that the book and some of the pictures show you one layer of peptidoglycan. And in reality, the peptidoglycan layer of a gram-negative cell is at least 3 layers of peptidoglycan, up to about 20 layers of peptidoglycan. And right here, you can actually see, I can blow it up. You can see there's several layers of peptidoglycan in this. There's one there, there's one there. There's another one there, there's one there. Any question about any of that? But the gram-negative cell is different in that the peptidoglycan layer is much thinner, and there's other molecules in the cell wall, mainly the lipids and carbohydrates, shown here and here. After bacteria have been stained with the crystal violet, all the bacteria will stain purple, either the gram-positive or gram-negative cells. And then Graham's iodine is used as a mordant. It will chemically combine with the crystal violet to form a larger complex, the crystal violet iodine complex. Next, the cells are treated with an alcohol acetone solution, which is called the decolorizer, and it will pull the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells. And why is because the negative layer of peptidoglycan, the thinner layer of peptidoglycan, just does not retain the crystal violet iodine complex, as well as the thicker layer of peptidoglycan in the gram-positive cells. So the gram-negative cells decolorize quicker than the gram-positive cells, and the timing of the decolorization step is critical. You have to time it correctly to make the reaction happen, and I'll talk a little bit more about that. You then apply the counterstain, which is safranin. A counterstain is just a different stain than the primary stain, which is the crystal violet. And the counterstain is a different color, and it will stain the gram-negative cells pink. It actually colors all stains pink, but we'll talk about that in a little bit. Now, there are some cells which give a variable result for the gram stain. And these are always the Mycobacterium. They have a slightly different gram-positive cell wall than the rest of the gram-positive cell wall. The Mycobacteria do not take up the gram stain very well. They do take it up, and if the gram stain is done correctly, they will appear gram positive. If it's done incorrectly, they will appear gram variable. And what is done incorrectly here is probably that the cells are too old to do the gram stain appropriately. You should be using cells that are only 2 days old or younger. What do you notice different about this pinkish cell here and that pinkish cell here? And it differs from this purple cell there and that purple cell there. What do you notice different about them? Nobody notices any differences?
Is it that they're just alone or not like piled with uh other ones?
Um, well, that one is actually next to another cell there. This one is next to another cell there too. I'm not sure what you're referring to. Maybe you're counting this one here as two cells. That's actually one cell there. And there's like a break right there in the cell. Let me shrink that down. It'll be a little more clear. There's like a break right there in the cell. And this cell looks like a ghost. These cells, on the other hand, as well as that one, look very distinct. They're very dark. There's very clear boundaries around the cell. And this cell has not very clear boundaries. That cell is like a ghost. There's no real clear boundaries. What's happening is these cells are dead. That's why that cell looks ghostly and why that cell looks like it's broken. These cells are dead, and the dead and dying cells are staining pinkish, or maybe they're just all dead. And then the cells which are viable are staining purple. And that's because the dead cells are not retaining the crystal violet, and then the viable cells are, and that's why these mycobacteria cells here look gram variable. But if you were to do this and you only grow the cells for 2 days or less, even if you're staining mycobacteria, they will stain gram positive. The only time I've ever seen some students who had gram variable results, they were staining mycobacteria, meaning that family of bacteria, and I asked them, how old are your cells? Because some of them are looking gram positive and others looking gram negative, and they always told me, oh, these cells are older than 2 weeks old. And I told them, well, that's not correct. To do the gram stain, you should only do it on cells which are 2 days old or less. The gram stain needs young growing cells for the stain to stain correctly. If the cells are more than 2 days old, some of the cells will be dead and they may not stain correctly. Any question about any of that? All right. So some tips for doing the gram stain. Let me blow that up so I can actually read it. Avoid using old cells. That's the number one tip. Prepare a thin smear. If you have a thick smear, it'll have trouble getting the crystal violet all the way through the cells down to the bottom cells. And then for decolorizing, it'll have trouble removing the crystal violet from the bottom cells as well. So always have a thin smear to make a bacterial smear correctly, you should only have the cells one layer of cell thick. And if it's not thin, the staining may not be not correct. Heat the bacteria on the slide only after the smear has air dried. If you heat the cells while they're still liquid on the slide, the liquid will boil and that will create holes in the bacteria cell wall, so the cells may not stain correctly. And then avoid overheat fixing the cells. That will disrupt the cell wall and actually makes the cell look like they're melted. So don't overheat the cells. Use fresh staining reagents and do not over decolorize, do not under decolorize as well. And we'll talk a little bit more about that. Any questions about the gram stain? All right, the acid-fast stains, the next cell we're going to talk about. Some bacteria, such as the Mycobacteria and the Nocardia, these are two families of bacteria, have a waxy substance in their cell wall called mycolic acid. Particularly the Mycobacteria have a lot of mycolic acid in their cell wall. So they are gram-positive, but they have an extra layer or structure in their cell wall of mycolic acid. And these bacteria are able to resist the acid alcohol decolorization in the acid fast stain. And we call these cells, the Mycobacterium and the Nocardia, acid fast positive bacteria, as opposed to acid fast negative bacteria. Initially, all cells will stain with the primary stain, which is carbol fusion, which is a cherry red color. We then apply the decolorizer, and that will use acid alcohol and remove the red color from the acid fast negative cells, but not from the acid fast positive cells. You then use either methylene blue or brilliant green as the counter stain to stain the decolorized cells. And then the acid fast negative cells will stain either blue if using methylene blue or green if using brilliant green. So the acid fast positive cells are reddish or pinkish, and the acid fast negative cells are greenish or bluish. Any question about that? All right, let's talk about the special stains. The special stains are a stain to stain a special cell structure within the cell or outside the cell. You can use the negative stain as a special stain to stain the glycocalyx. If the glycocalyx is firmly attached to the cell wall, we refer to this as the capsule. You can see the capsule under a light microscope. Let me blow this up a little bit. So there's the cell membrane. There's the cell wall peeled away from the cell. So right there is the cell wall. And that greenish layer will be the capsule. The capsule is external to the cell, something secreted by the cell. It surrounds the cell. And if you can see it under the light microscope, it is called the capsule. If you can't see it under the light microscope, that means it's mostly water, it's loosely organized, and it's called the slime layer. You need an electron microscope to see the slime layer. Both the capsule and the slime layer are easily washed off the cell by normal staining techniques. So in the gram stain, you'll wash the capsule off and you will not see it. You have to use a special staining, gentle staining technique to see the capsule. and you can only see the capsule under the light microscope. You can't see the slime layer. The production of the capsule is important because it can be associated with the organism's virulence or its pathogenicity. The capsule protects the cell from being phagocytized. Why Streptococcus pneumoniae is such a dangerous disease if somebody got it, especially if they got it before the development of antibiotics, because the bacteria has a capsule which protect the bacteria from the body's defenses, meaning the immune system had a hard time fighting off Streptococcus pneumoniae because the cells have a capsule. Another example is actually strep throat. Streptococcus pyogenes has a capsule, and that's why it is hard for an individual to throw off strep throat. The immune system has a hard time attacking it because it has a capsule. And so you really need antibiotics to throw off strep throat. It is possible to throw it off, but many people do not throw it off. And that's why it develops into strep throat and then can go on. And it used to cause many different serious diseases like, see, oh God, what was that name? Not rheumatic fever, but something like red fever. Scarlet fever, that's it, scarlet fever. Could be a disease that would develop after strep throat. Scarlet fever is now very rare because of antibiotics, but it is a serious disease. Some organisms have this capsule and it tends to make the cells more pathogenic. Here we're seeing cells with a capsule, and you can see the colonies are mucoid looking. They're covered in mucus, and that's because the cells have a capsule. Streptococcus pneumoniae is actually grown here, and it looks mucoid because it has a cell capsule. A negative stain technique is used to show the capsule under the light microscope because it's a gentle procedure using India ink or nigrosin, and you just put the cells in it, and there is no washing. With the India ink, it stains the background very dark. It stains the cell a very light color, and then the capsule is unstained. Here is probably using nigrosin. This might be using two stains, one to stain the cell, one to stain the background. The background is colored. The cells are darker colored, and then the capsule is unstained. This is Klebsiella pneumoniae using India ink here. All right, the endospore stain is a stain looking for endospores. They're a metabolically inactive, hardy structure produced by some bacteria, and they are a defensive strategy for the cell to survive an unfavorable environmental condition. When the environment becomes favorable, the endospore can then germinate, and then the bacteria will return to their normally metabolically active vegetative state. So the vegetative cell, the parent cell, is active, metabolically active, reproducing. It has its normal shape. And when the environment becomes deteriorating, the parent cell can give rise to an endospore. The endospore will survive the unfavorable environmental condition. The endospore has very low metabolism going on, if any metabolism, and it's inactive, it's not reproductive, and it usually has a different shape than the vegetative cell. Most endospores are made by two families or genera, the family Bacillus and the family Clostridium. These are both genera as well as families. There is one or two other families that make endospores, but they only have one member in the family. And the Bacillus has many species. The Clostridium has many species. So most of the species that make endospores belong to the family Bacillus or the family Clostridium. This table is showing you some of the endospore forming bacteria that are pathogenic, meaning the endospore forming bacteria that can cause human disease. Bacillus anthrax, which causes the disease anthrax, has an endospores. Bacillus cereus, which causes food poisoning, has endospores. Clostridium botulinum, causing food poisoning, meaning botulism. It has a endospore. Clostridium tetani, which causes tetanus, has an endospore. Clostridium perfringens, which causes gangrene and food poisoning, also has an endospore. And then C. diff, Clostridium difficile, which causes antibiotic-associated colitis, also called pseudomembranous colitis, also has an endospore. And that's why C. diff is so difficult to cure. You have to treat the patient 2 times with antibiotics in order to successfully cure a C. diff infection, because the first time you treat it with antibiotics, the endospores will survive, and then you stop giving antibiotics, the endospores will germinate, and the person will re-establish their C. diff infection. We use the Schaefer-Fulton endospore stain to stain for endospores. It uses malachite green as the primary stain, staining all cells green. It is then applied as the mordant. It helps the primary stain get in the endospore. The cells are then decolorized with water. Water removes the malachite green from the vegetative cells, but the endospores retain the malachite green. Safranin is then applied as the counterstain, which stains the decolorized cells. So the vegetative cells will stain pink from the counterstained safranin. The endospores will stain green from the malachite green. Oops. So here we're looking at an endospore. This endospore has come out of the cell. You'll note it's Coccobacillus in shape. The pinkish cells are the parietal bacteria cell or the vegetative cell. And there we have an endospore being made inside the parent cell. You can see the red right there on one end of the cell, and then the bigger red on this end of the cell, and then the endospore being formed close to the middle of the cell. It's actually off to the side of the cell. And there's another one there. Why it's called an endospore, it forms within the parietal cell. All right, so an endospore is a structure to help the cell survive. It is not a reproductive structure. Any question about the endospore staining? If not, let's talk about the flagella staining. The flagella is a long whip-like appendage to cells, providing bacteria cell as well as archaebacteria with their motility, meaning if the cell can locomote or swim in the liquid media, the bacteria have at least one flagella. Flagella do not stain with normal staining procedures. So with the Gram stain, you will not see the flagella on the cell. Flagella are composed of flagellin, which is a class of proteins. You don't need to know this for a quiz, but for this lab, you need to know the distribution of flagella give the cell its flagellar arrangement. If there's one flagella on one end of a cell, we call it a monotrichous cell, one flagella. If there are multiple flagella on one end of the cell, we call it lophotrichous. If there are multiple flagella on both ends of the cell, but only on the ends of the cell, we call it amphitrichous flagella. And then if there are flagella all over the cell, in different regions of the cell, we call it peritrichous. So the flagella are too thin to be seen with standard staining techniques. So you require a special stain in order to see them. The flagella stain normally coats the flagella many times so that you can see the flagella. And these cells have been stained for the flagella arrangement. You can note it looks like that cell has no flagella, or it might maybe one. That cell might have no flagella. That cell here has one flagella right there. That one looks like it has two. This one looks like it has two. That one might have flagella on both ends of the cell. It definitely has one on this end of the cell. And I'm not sure if that's coming around from the other side or what, but it looks like there's a little bit here, which looks like this bends around here. So that has flagella, multiple flagella on both sides of the cell. And then we have peritrichous cells on this side, on this cell, and on this cell and that cell. So why do we have cells with multiple flagella arrangements in this picture? Anyone have a guess? All right. Well, initially, when the cell first divides, it will have no flagella, which might be that one or that one. And then the flagella will be made. Initially, the cell will put out one flagella on one end, which I'm having a hard time finding one here with just one. There's maybe one right there. And then the cell will put out more flagella, usually on the same end. Like this one has two on this end right there. And then it'll have more flagella on both ends. And then it'll start putting flagella out all over the cell, and that will be peritrichous. So these cells are peritrichous. The difference is some of the cells just haven't put out all the flagella. Some of the cells only have one flagella on one end, and some of the cells might have no flagella. So it just depends on where you're catching the cell and its ability to make the flagella. And eventually this cell here, assuming it lives, will become peritrichous. And so we call this a peritrichous. You always call it by the highest number of flagella you see. You do not call the cell all the possibilities. Let me see if I can shrink this down and go back. So you would not call this peritrichous, amphitrichous, lophotrichous, and monotrichous, as well as no trichous. It's simply peritrichous. Any question about that? All right, you should know the terms of this lab. References you can skip. Well, you're not physically going to be performing any of these activities. Everything will be online, although we do have a virtual lab today. You must understand how to prepare bacterial smears for staining, the principles and procedures used in the various staining techniques, and be able to interpret those results using microscopy. So on the first thing, we're going to prepare a bacterial smear for staining. So the bacterial smear is done and is independent of the staining. We'll also look at how to heat fix the cells. So there's the steps right there. So watch the video and then watch. You need to air dry the cells and then heat fix them. Which actually that's right there. And I should have just read it because it's so little. All right, procedure 2. You're going to be looking at the simple stain. There is a video clip of the simple stain. That's when you simply add a single dye to the cells. Wash the dye off, let the slide dry with bibulous paper, and then look at the cells under the light microscope. So you note that the bacterial smear is something you do before you do the staining. So it's independent and separate, but it must be done before you do the staining. Here we're looking at some cells that have been simple stained, and the Staphylococcus aureus and E. coli. Let me warn you, this picture is a bit grainy, and it looks like the cells are pleomorphic or something. And you'll note that that right there is one cell. That's one cell. Right there is one cell. There's one cell. So this is not a pleomorphic cell. And this was actually one there, one there, one there, and one there. So that's actually 4 cells together. That right there is 2 cells together. One there, maybe 3 cells. One there, and then one there. There's 2 cells together. So make sure you get the shape right here, because this is not a very good picture. And then describe the cellular morphology of E. coli and Staphylococcus aureus. What's the total magnification of the slide here and here? It's labeled above. And then on B, tell me what the total magnification is. All right, procedure 3, we're going to look at the gram stain. There's a video to watch on the gram stain, and then you're going to perform the virtual lab on the gram stain. See if I can get that to go. Let's go that way. Get it to run. Hit continue.
Hi, Lab Assistant.
I'm just going
All
to move
yogurt
forward to the actual steps of the
Unfortunately,
gram stain.
This is
You
what.
can
Now
read
take a look
through
at this yogurt. Look,
what they're
let's
doing.
in the gram staining process. Bacteria. We are going. All right. To prevent bacteria from, you're going to use a striker to light the Bunsen burner.
Wherever you have
The
the
gas is
arrow,
turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
how to do this is you pick on what you're supposed to put your mouse over and then bring it down in the shape of the arrow. And that
We need to
what
dilute the
works it.
yogurt before we put it on the mic. Start by placing 1 milliliter of the diluent into it. This is an inoculation. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. After it has cooled down, use the loop. Place the yogurt sample into the previous. Now, use the inoculation. Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it direct. Transfer a small amount of sample from the test tube to your prepared slide. Place 2 loops full from the sample onto the slide. Tilt the slide to spread the drop out slightly, about the size of. Now we'll heat fix the slide. Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Now we begin the staining process. We use 4 chemicals to stain a slide.
All right,
We
here's
apply
the
them in
gram
a
stain.
specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of grams iodine. Squeeze several drops onto the sample, enough to let that sit for 60 seconds. Now rinse it under slow running water. Now we are going to use the alcohol solution. This will wash off the excess dye, leaving only the dye fixed to the bacteria.
A better way of applying the alcohol is to actually hold the slide up and then put the decolorizer on one end, the top end of the slide, let it run down, and then you keep adding decolorizer until the blue stops running on the slide. Once the blue stops running, stop adding decolorizer.
Use the alcohol dropper to cover the sample. Let the alcohol sit for 30 seconds. Time is very important in this step. Don't let the alcohol sit for more than 30 seconds.
All right, they're using 30 seconds because they're only using alcohol in most of our lab, as well as in our lab at Clark College. We use acetone alcohol or alcohol acetone, which is stronger, and it's only applied for 5 seconds. But the 95% alcohol is weaker, and that's why it's applied for 30 seconds.
Now, rinse it off under slow running water.
All right, I
You
think
now
that's it for
use the dropper to cover the slide. Let that sit for 60 seconds. Now, rinse it under slow running water. Now our slide is ready, but gently press the paper down a few times to blot the water off the slide. Good job! You now notice the timer and the 2 buttons next to it. These buttons let you decide how long to wait for. Remember, you can tap.
All right. Any questions about that video? Like I said, you'll have to go through it on your own. So the steps used when performing the gram stain: first, flood the smear with crystal violet. Let the crystal violet sit for 1 minute. Then you pour off the crystal violet and do a wash. In between each step, you do a wash. Second, flood the smear with iodine. Let it sit for 1 minute. Pour it off. Wash with distilled water. And then you flood the smear with alcohol acetone. This one you're going to let for 5 minutes. Like I said, the video was only using 95% alcohol, in which case it's applied for 30 seconds. When using the alcohol acetone, it's best to hold the slide up and then view the slide at an angle. Add the acid or alcohol acetone decolorizer, let it run down, and just keep adding it until the blue stops running. It should stop running in around 5 seconds. Once the blue stops running, you stop adding the decolorizer. And then you very quickly wash that slide with distilled water. You want to remove the decolorizer as soon as possible, so you wash it right away. And then you add the saffron in for 1 minute. And then you pour it off and then wash the slides with water. And then you blot dry the slide. So you have bibulus paper. You rip out a sheet. And then blot it down. I don't know if you can see this. Hopefully you can. Blot it down. You do not move the slide underneath the bibulus paper, nor do you move the bibulus paper on the slide. You just blot it down, and that will remove the water. It will not remove the cells, or at least most of them. And it will remove enough water so then you can use the microscope, including the 100x lens with oil. Any question about any of that? So when you're giving me the steps of the gram stain, you do not need to tell me the water wash after each steps, except for maybe the first time you should say, and then water wash. And you can make a comment and then do this after every next step. So here's looking at the cells. After the first step, after applying the crystal violet, all cells will appear blue-purple. You then add the Graham's iodine, and all cells will stain a little bit darker, a little more purple color. The cells do have a larger molecule. The crystal violet iodine complex is larger than the crystal violet or the iodine alone, and that'll help the cells intensify the color in the cell, as well as it's a slightly different color because the Graham's iodine is a little different color. You then apply the decolorizer, and the gram-negative cells will appear clear. The gram-positive cells will appear purple as long as you're doing the gram stain correctly. If you apply the decolorizer for too long, you will pull out the crystal violet iodine complex from the gram-positive cell as well as from the gram-negative. On the other hand, if you apply the decolorizer for not long enough, you will not pull out at least all of the crystal violet from the gram-negative cells. So the decolorizing step is critical. And like I said, the best way to do it is to hold your slide up and then apply the decolorizer. Once the decolorizer stops running blue, stop adding decolorizer. And then you immediately wash the cells with cold water to remove the trace amounts of decolorizer that remain on the cells after you pour the decolorizer off. You then stain with safranin. Safranin actually gets in all cells, but it will make the gram-negative cells appear pink and the gram-positive cells will appear purple. Any question about any of that? All right. Here's a chart, 2 smears with the gram stain. The E. coli state whether it's gram positive or gram negative, and then give me the cellular morphology. Do the same for Staphylococcus aureus, whether it's gram positive, gram negative, and the cellular morphology, whether the cell is coccus or bacillus. Any question about any of that? All right, then for procedure 3, answer procedure 3A. Staphylococcus aureus, after the primary stain is added, will be what color? Pink, purple, or colorless? Just come up to place here. It's in the reading there. Then answer question 3B. E. coli, after the primary stain is added, is which color? For question 3C, Staphylococcus aureus, after the alcohol acetone step, is what color? Give your answer there. For D, Escherichia coli, after the alcohol acetone step, is what color? For E, Staphylococcus aureus, after the counter stain is added, is what color? And then E. coli, after the counter stain is added, is what color? Any question about any of that? All right. If this is correct, why did I say the safranin goes in all cells, meaning it does go in the gram-positive cells, but the gram-positive cells are not staining pink? Why is that? Nobody has a guess? Well, if you were to look at the gram-positive cell under the electron microscope, you would notice that some regions of the cell are pink and other regions of the cell are purple color. Which that's not purple. I guess it should go there. It's just that the purple color to our eye is more visible and dominates over the pink color. So with our eye, we cannot see the pink color in the gram-positive cells. We only see the purple color. To see the pink color, you have to use an electron microscope and blow the cell up many times. Any question about any of that? For E, Staphylococcus aureus after the counter stain is added. For F, E. coli after the counter stain is added. So tell me the color. In procedure 4, we're going to look at the acid-fast stain. There's a little video clip on the acid-fast stain. Review the steps used when performing the modified Kenyon acid-fast bacteria stain. We first add carbol fusion. It gets in both the acid-fast positive cells and the acid-fast negative cells. We then use the acid alcohol wash. It's the decolorizer. It will pull the carbol fusion from the acid-fast negative cells, and they will be colored clear. It will not pull the carbol fusion from the acid-fast positive cells, and they will appear red in color. You then apply brilliant green as a counter stain. Look at the cells, and in this case, we can see carbol fusion positive cells, meaning the acid-fast positive bacteria, stain reddish. The acid-fast negative cells stain greenish from the brilliant green. Any question about any of that? Carbol fusion is applied for a longer period of time. You need to apply it for 4 minutes. Then wash off with water. Decolorize with acid alcohol for 10 to 15 seconds until you can no longer see the pink running freely off the smear. Gently wash off the acid alcohol with tap water. Take off the excess water, and then you stain with brilliant green or methylene blue as a counter stain, and then gently rinse the slide with water. Get the worst of the excess water off the blot with bibulous paper or blotting paper. So once again, you get your slide, rip off your bibulous paper, and then blot the bibulous paper over the slide. Any question about any of that? And then interpret whether the organisms denoted by an arrow are acid-fast positive cells or acid-fast negative cells. So there's one arrow there, one here, and one there. So that cell there, is that acid-fast positive or acid-fast negative? Well, that cell there, acid-fast positive or acid-fast negative? Not hearing any answers, people. Well, you can answer the arrows yourself and then turn in the lab. Procedure 5, we're going to look at the special stains. We have a video clip on the endospore stain and a video on the negative stain. So 5A applies to picture A, 5B applies to picture 5B, question 5C applies to picture 5C here. So A, this is the Schaefer-Fulton stain, which uses wet stain as the primary stain and ultimately dyes which structure, which are produced by some bacteria to aid in survival. So fill in the answers. And then 5, I guess this is 5A1, 5A2, lists some examples of pathogenic endospore producing bacteria. If you need to, you can get the answer from the table in the lab module. For procedure 5A, let me go to the worksheet in case I haven't changed the questions. For procedure 2, is that where we are? Tell me the cellular morphology of E. coli and the cellular, yeah, we're past here, sorry. And then for 5A2, what are some examples of pathogenic endospore producing bacteria? It is numbered or given in the table in the lab manual. For procedure 5B, this is a stain for B that coats long whip-like appendages which aid in motility. State the flagella arrangement of the cells in this picture. And then for B3, what are some examples of clinically significant bacteria that produce flagella? Everyone who looks up the answer should get it right. Do realize it asks for examples. So make sure you have at least 2 examples. If you only have 1, you only get partial credit. 5C, this is what stain that uses India ink or nigrosin and can be used to detect the presence of what cell structure, which is located exterior to the cell wall and helps prevent phagocytosis. C3, what are some pathogenic microorganisms that produce a capsule? You should be able to find that in the lab, in a table in the lab module. Any questions about question 5 or the lab so far?
I have a quick question for
Uh-huh.
these last A, B, and C questions. You said we can find it in the lab module. Would it also be found in the lab book
You've
or
got the lecture textbook.
well, both?
We don't have a lab.
The app,
Oh, the atlas. If you have
atlas.
the atlas, yeah, you could find it in the atlas. You could also find it in the lecture textbook, I believe, and certainly
Okay,
in the lab module.
perfect. Thank you.
All right. 5C, this is what stain that uses India ink or nigrosin used to detect the presence of a cell structure. Name the cell structure, which is located exterior to the cell wall and helps prevent phagocytosis. And then 5C3, what are some pathogenic microorganisms that produce a capsule? I've already told you the 2 that I know, but you can list any that have a capsule, and as long as they're pathogenic, it would be correct. All right, then we have two case studies. Case study 1, a 45-year-old female is experiencing some respiratory issues. Based on her clinical presentation, the doctor orders a chest X-ray and a sputum for acid-fast bacteria culture. In addition to the culture, an acid-fast beer smear is made and stained. Based on the findings below, what is the most probable diagnosis? You must name the species which causes the infection. So there is the x-rays of the lung, and you can note the lung looks really weird, especially in this region. The lung should be totally dark, sort of like this region here. And obviously that region is not correct. This region is not correct. That region up there is not correct. And then we have the sputum smear, and then the Zeal-Nelson bacteria stain. Any questions about case study A? Remember, you must name the species. You cannot just simply tell me the disease. In case study 2, we have a 64-year-old diabetic patient admitted to the hospital with soft tissue necrosis. You can see it here. A routine culture and gram stain is ordered. The bacteria grew aerobically. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing the infection. So there's the gram stain there. You can see the gram positive or negativeness. You can also make out the cell shape. And perhaps can make out the what that is called. We'll just say make out the patient's symptoms and give me the name of the bacteria causing the infection. You must name the species causing the infection. And there's the Schaefer-Fulton stain showing something in the stain. Any question about the worksheet or the lab module? If not, I'll be here until 8 o'clock or until the last student logs out. Go ahead and work on the lab. Remember, I'm here to help you if you have any questions. Some of you are getting confused on the question, and so you should ask me for clarity. Some of you just need a little help, and I'll try and help you.